Welcome to Spark Science, where we explore stories of human curiosity. I'm Regina Barber DeGraff. Today we are in the Digital Media Center here at Western Washington University, and we are talking with my good friend, Dr. Robin Kodner, and we're going to talk about Ski2Sea algae. So welcome to our show.
Thanks for having me on, Gina.
So I wanted to bring you on mostly because I've been meaning to for a while now. Lena, our collaborator, has been on the show, but you do amazing work with like algae in the mountains and in the bay. So that's why we're calling it Ski2Sea algae, this show. So can you give us a really short description of like what you do and your position here at Western?
Okay. I'm an assistant professor in the biology department, and what I do for my research is I'm interested in microbial communities and what diversity of organisms there are in those communities and how they change over time. And so I started off studying them in the bay, so I mostly worked on marine systems.
Here in this bay, or many, like in your undergrad,
well, I've always worked on algae, but I started doing this kind of work when I was a postdoc at the University of Washington, and so I was working in this region, in the Salish Sea, and I specifically started working on Bellingham Bay when I moved to Western. And then through that project, I realized that I was actually also very interested in algae that live on snow because I spend so much time in the mountains, and that there hadn't been very much work done on the algae communities that live on snow, especially using the modern DNA sequencing techniques that I use.
Real, so there's not a lot of people like going up to
very
mountaintops.
Little, and in fact there's no publications at all on using these new DNA sequencing technologies for characterizing the communities in the North Cascades, or really the whole Cascades. So,
or any other like mountainy, snowy.
So people have just started to publish on this work, and there's like a paper from Russia, and there's a paper from like Svalbard, like up in the Arctic in Europe. And then there's a lot of work done in Antarctica. Actually, shockingly, there's more work done on Antarctica than there is in the North Cascades.
Which is maybe easier to get to.
A little easier, right? As if you just drive up the road for an hour and a half and you can be there.
Right.
From Bellingham.
Right, you were just there.
I'm in the mountains a lot.
Yeah,
so
very recently.
I originally started doing this work in marine systems, but now I've been putting a lot of effort into studying them in the mountains.
Yeah, so I just want to bring this up because I like to use the beginning of our show to kind of humanize scientists because we're very scary to people sometimes.
Yeah. I'm especially scary to people.
Are you?
Very scary.
But you, Lena's another biologist here at Western, you both were in a Ski2Sea team last year. Let's tell our listeners and our viewers about what Ski2Sea is, because I just realized that not everyone that's going to be listening is from Bellingham, knows what this big like event every year is. Right. So let's give them the gist.
So Ski2Sea is this relay that goes from the Mount Baker ski area to Bellingham Bay. And so it's a big event that happens every year, and it's like this big thing in Bellingham. Like a lot of people have done it for years and years and years. And
all the hotels are like filled up.
Yeah, there's all this tradition in it. And so when I first moved to Bellingham, I didn't do it my first year, but then I felt really left out. And so I've done it every other year because
I grew up here and I've never done it.
It's this thing. So
I'm not athletic.
Yeah. So Rachel Severson and I decided to start this Whatcom Women's team. And we pulled all these other Whatcom Women Scientists team, and we got all these other women scientists to join us. And what was really cool is we weren't all professors, we were scientists in all these different ways. And so we had a geologist who was like a working geologist, we had a fisheries biologist who does consulting work, we had a couple of professors, we had a grad student, so we had a mix of different kinds of women scientists.
Can you tell me just a little bit about this as we're humanizing you?
Right, we were actually on the team together for a couple of years. The team was called the Whatcom Women Scientists, and a couple of years ago, there wasn't enough teams in the Whatcom Women's division to keep the division going, and so there was a big push by some leaders and leader athletes in the community to get a bunch of women's teams. And a friend of mine and I, another professor at Western, we went to the meeting and we decided to form a team, and then we pulled a couple more people into the team, and we were trying to figure out a theme for our team, and we realized we were all scientists.
What was your leg, and like, so it goes from the mountain and you're skiing, and then
so there's a cross-country ski leg, and then a downhill ski leg, then a road run that's in the mountains, so you like start running down the Mount Baker Highway.
Right, for how long?
8 miles, and then that person hands off to a road biker who rides for maybe 40 miles.
That's worse.
And then they hand off to a pair of canoeists,
Right.
canoeists, and they canoe.
Canoeists.
They canoe.
From their Church of the Canoeing.
They canoe on the Nooksack.
Right.
And they hand off to a mountain bike / cross bike leg that goes through Ferndale down to the water, and the cross bike hands off to a sea kayaker.
Right.
And the sea kayaker finishes the.
And that's where I was, and it was so loud, like, so maybe the audio won't be great at all.
And it's really exciting. And so when you start at the beginning, everybody's all like at the cross country ski, like all 500 teams or so start at once and then get spread out. And so by the time you're at the kayak leg, teams are pretty spread out.
Right. And you won a medal, right?
We sure did. The year before, we missed getting on the podium by 2 seconds, and it was my fault. I got passed in the last 2 seconds of the last leg.
So what was your leg? You didn't ask. Answer.
My leg. So I did the cross bike
Okay.
almost for 2 of the 3 years that we did the team.
Okay.
The 2015, it didn't snow very much. There was no snow at the ski area,
Right.
so the legs got moved around a little bit, and there was a mountain bike leg that ended the race, and I'm a mountain biker, and so I did that leg.
Okay.
And that's when we lost by 2 seconds because I got passed at the very end.
So you were like, the next year you needed to get those extra 2 seconds.
I did, and so I was doing the cross bike, and I was like determined to do well. I'm not super competitive when it comes to sports, and I mean I like to do. I just am not like into racing so much, but this year I was like, I'm gonna redeem myself from the year before.
Right.
And so I was in really pretty good biking shape, so I thought, all right, this is the year. And then
You'd been planning for a year, so
know.
yeah.
And then in my race, I got a flat. But it also takes some minutes, and
Right.
like minutes matter in the race. And so
Yeah.
I rode on a flat tire for like 10 minutes.
Wow.
On rough terrain. That was my decision. I don't know if it was the right one.
But you got on the
But
podium.
we won. We still won 3rd place anyway, despite my slowdown. So we would have like really kicked it. Although some of the other teams in the Whatcom women's division are really competitive, and so they're really, really
So they like
amazing
slashed your tire?
athletes. No, they didn't need to slash my tire to win. But I feel like we sort of won in some ways of the rest of the crew that weren't like professional athletes.
Oh, that's awesome. Well, I mean, I really love that story. So we're kind of talking about this idea of algae up on the top of the mountain, algae in the bay, but I want to go back to how did you become a scientist? Because we're talking about like this women's science Ski2Sea team. You know, Wonder Woman just came out. Like, I want to talk about this idea of kind of being inspired to be in a field that women aren't really populated in, and that's science.
Yeah.
And biology, it's a little kind of complicated, but still there are a lot of inequities in many things. So I want to talk about that.
That's true. And in my training, so when I did my PhD, I was actually...
And where did you do your PhD, Robin?
I did my PhD at Harvard.
Yeah.
You might have heard of it?
That's one of the only schools I heard of when I was growing up.
Oh, that's right.
That and UW.
I did not come from an academic family, so I never in my wildest dreams ever imagined I'd end up at a place like Harvard.
Yeah.
And this is what I always tell people, especially my students, is when I was an undergrad, I feel like my strength as a scientist is being creative. Like, some people are really good at attention to detail and being very organized and like knowing all the nitty-gritty. Like, I'm not very good at that. But I'm good at like having a big picture idea and being creative and sort of bringing things from different fields together. Like, that's what I like, and I was always like that. Even when I was an undergrad, and I actually have another amazing woman to thank for that. She was my best friend all through growing up, and she's now a doctor, physician in San Francisco.
Awesome.
And she and I were undergrads, I think it was our sophomore year, and I had taken honors organic chemistry, and I was like, I'll never take another honors class again. Like, forget it. It was horrible. Yeah. And
sometimes that happens.
We were both in going to take biology, and it was like the deadline to sign up for the honors section. And I was like, I already said I'm never doing another honors class. And she convinced me to sign up for the honors section because if you did the honors section, you got to do an independent research project instead of doing the regular modular lab like everyone else had to do, where you have to go in and just like follow the lab book.
Right.
Which recipes, which of course I hate because I don't like the details. I had to find an advisor really quickly, because I like decided to do this on Friday at like 3 o'clock.
Right.
And what's amazing is that I went, and
Like
this was,
every student.
Right.
It's not just you.
and this was in the 90s when, you know, search engines were pretty new. And I was at the University of Wisconsin in Madison, and I went to the research webpage, and I had to like quickly think of something, and I was like, evolution. That's what I want to study. So I type in evolution, and I press return.
Super generic.
Right, like any good sophomore in college would do. And there was 2 people who came at 2 hits.
Well, that narrowed it down at the very
Totally
least.
narrowed it down. And what was shocking is that both of these professors were in their offices on a Friday afternoon.
Wow.
And talked to me.
That's very lucky.
And the first one was this guy who studied the evolution of physiology in mice. So he had a mice lab. And at the time I was like, I don't think I could have done that.
Yeah.
And then I went to the second person, this woman, her name is Dr. Linda Graham, and she studies the evolution of the transition from algae to plants. And I went to her office, and she was super friendly, very welcoming. And then she said, well, how would you like to boil moss in acid?
You're like,
I was like, that sounds great.
Yeah.
I'll do that. And
you're you're a vegan. You're like, I love plants. Let's do this.
Yeah. And I started working. She was interested in seeing what parts of mosses would potentially preserve in relation to algae structures. And I started working in Linda's lab, and she was an incredible mentor to me. And I had an idea about a senior thesis project I wanted to do, and I needed some help from my geology professors, and they all kind of like patted me on the head. They're like, oh, you're so cute. This is good. This is such a cute idea. Okay.
What, so you were in geology at the time?
I kind of was in between geology and biology because I was interested in these like evolutionary questions that would leave a fossil record. So I took a lot of geology classes. And even though biology is a lot of women, geology maybe less so. So
yeah. Still better than physics.
Yes, still better than physics, but I was sort of in between there. And so my professor supported this funny idea I had for my senior thesis project.
And what was it? What was your idea?
I wanted to look for early land plant fossils in the rocks around Madison, Wisconsin.
What kind of plant is that?
It would be kind of like a moss.
Okay.
Or a liverwort, if you know what one of those are.
No, God, no.
Liverworts are so cool. They're real simple. They're like the most simple plant. Anyway, there's lots of them in Washington because they can only live where it's wet all the time.
Okay.
And anyway, I got to publish that research in Science, the journal.
As an undergrad.
Right.
So for our listeners and our watchers that don't really understand the magnitude of that.
That was a big deal.
For one, not a lot of undergrads get to do research, period. I mean, if you were to go to the University of Washington, these big R1 schools, it's very, very hard to get any sort of, you know, real science or research experience as an undergrad. If you come here to Western, I'm just going to quick plug for us, there's a lot of research that you can actually do as an undergrad. You have some undergrads and grad students working for you, but on top of that, to get whatever you're doing in undergrad to actually get published is huge. That's like another tier. And then to get that in Science magazine is like, and
it's pretty mind-blowing.
And then you go to Harvard.
Then you go to Harvard.
And you go to
Harvard.
There you go. That's
it's
how
funny,
it happens.
Because I was so naive when this all was happening. Like, I was just psyched about my research. And my Linda Graham, who
and it was your idea, so that's why.
It was my idea. I really give a lot of credit to Linda Graham because she really encouraged me and supported me.
Right.
And, you know, every good scientist has a mentor like that somewhere in their history. And she was like, I think we can send this to Science.
Yeah.
And I was like,
whatever that is.
Sounds great. We're doing science.
I
didn't know the difference. And
I didn't either at the time.
We wrote another paper that we put in the American Journal of Botany, I think, which is also a really great journal for plant science.
It ain't no science.
But I didn't really know the difference between the two. Like,
Right.
it didn't mean anything to me. And then everyone in the department in Madison, like, freaked out.
Right.
Because they're like, we have an undergrad who just published in science. But I still didn't quite understand.
Right.
Until I went to grad school.
Tell us about, like, what do you do on the mountain and how does that relate to the algae that you collect in the bay? Like, what is the connection between all of this stuff?
So the connection between all of this stuff is really the methods that I use. So I use these, it kind of falls into the field of environmental genomics. So
sounds intense.
DNA sequencing technology improved dramatically about 10 years ago. And we're able to sequence tons of DNA really cheaply. And that allowed us to people like me who are doing ecology with microbes to kind of go to an environment, scoop up all the DNA that's there, sequence it, and then we have like millions of tiny pieces of DNA that we then put back together in the computer, and we can say something about the community. And the reason we have to do that is we can't see our organisms. So if you wanted to describe what was going on in a forest, you would just go to the forest and you would look around and you would count the plants that you see and count the animals you see, and it's pretty easy. We're kind of doing the same thing, but with stuff that we can't see. And so the way that we see it is with DNA.
Okay.
So that's the link between the two. But the way that we collect the samples are really different.
Okay.
So on the mountain, we scoop up snow and we collect it into DNA preservative. And the other thing that we do is we can image our cells in the field using a field microscope. I have this
So awesome.
cool little field microscope.
How big is this field microscope? What does it look like?
It's like about this big. It fits in my hand, and
Okay.
it has like a single, has a bunch of objectives that are actually pretty nice. It magnifies 600 times.
Okay.
So it has a 60x objective and a little eyepiece, and you look into it.
Okay.
And what's really cool is the company that makes these microscopes designed them to be distributed in developing nations so they could diagnose malaria in the field. So you can see the malaria parasite in the blood if you have a nice microscope.
Wow. And this is just a handheld like device.
Yeah, and so it's really cool that that's why it was developed, but they're pretty inexpensive. And
Yeah,
so
how much? I was about to say, how much do these things cost?
about $500.
Okay.
So it's expensive, but it's not
For like
crazy.
lab equipment, it's
But
not
for lab equipment,
expensive.
and for microscopes, that's really cheap.
Okay.
And so you take a sample and you just put it on the microscope, and it's all newfangled technology. So there's a attachment for a smartphone.
Oh, wow.
So that you can then image what in the field what you're seeing, which is really great because it's one thing to like describe what you saw and try to draw a picture of it, but it's much better to just be able to take a photo. And I realized a couple weeks ago that then I could also take videos. So some of the algae that live in snow, it had been hypothesized, go through this life cycle change where they can actually swim through the snow. And no one had actually observed that directly in the field. There's been some observations, but I actually took a video of swimming algae in the snow 2 weeks ago.
Can you like publish that and be like, I was one of the first people to have like video of?
I think so, yeah. You put it in a supplemental when you send in your...
So is it on your website now, or no, that's like...
No, it's secret.
It's secret.
No, it's not secret.
Well, this isn't going to be produced for a little while, so maybe you'll be out by the time.
No, it's definitely not secret. I think I'm going to share a little. I think I'll share it on Instagram when I get a chance to like edit the video a little bit. But yeah, it's really cool.
Well, that's so awesome. So like, I was going to actually ask, this is an aside, we'll come back to your research, but you just made a really good point that I hadn't thought of until just now, and how the history of biology and being a biologist and field work and kind of categorization of species, there's been a lot of drawing. So like art and biology has had this huge relationship throughout the hundreds and hundreds of years, like Darwin drawing and all that kind of stuff, animal species. Now that we have phones, like, is that still taught? Do you know what I mean? Like, is that still taught in biology?
No.
Yeah.
That is actually an amazing question.
Thank
and
you.
It is not taught. And I just came back from an algae conference, and there was a woman there who received an award for art and science. And she, I wish now I had the book to bring, but
we can pretend they're these books.
I wish I had that book to share.
Yeah.
It is so cool. It's called A Secret Garden, or An Ocean Garden, The Secret Life of Algae. And her name is Josie. But anyway, she did this, like, she's done a lot of work to compare her scans and her photographs with these old drawings from the 1800s.
Oh, wow.
Where scientists were originally describing these species, and they did these elaborate drawings that are beautiful. So they're definitely art.
Right.
So yeah, there's a lot of that in
that's
in the
amazing,
history of algae.
She's double checking like what they were actually looking at.
Exactly.
What if they labeled it not correctly? I mean, they don't know, right? They didn't have a big enough sample, right?
Exactly. And now that we use DNA, we realize that a lot of those a lot of those species descriptions we made based on looking at the algae aren't actually correct, because the algae can change their shape really easily, so they can look really different and be the same species.
Yeah.
So there's also a really incredible story about an artist who works on an algae that I study that's related to Western.
Yeah, tell me the story, because this is
it's kind of an aside from my research,
that's
but it's
okay.
something.
We'll come back to your research, but this show talks about sharing stories of human curiosity. Let's do it.
So this is the reason that I actually came to Washington in the first place when I was at Harvard in grad school. So there's a sort of unique group of marine algae, microscopic, that paleontologists think is the oldest evidence for eukaryotic life, the oldest evidence for complex life. But no one had actually really studied it. Very few biologists have ever studied it. And so it was just kind of like this lore.
Conclusion?
Well,
They're
because
just like shared it.
paleontologists do this thing where they like find a fossil and then they try to compare it to living things and they just compare the like structure.
Okay.
And these things look like marbles. They're just little spheres without any structure.
Right, yes, okay. I remember reading about this.
They just compare these like, okay, this looks like a marble and this looks like a marble, probably the same. I mean, it's really that simple. So my whole dissertation was about trying to like really investigate that further using chemical methods and all kinds of things. But I really needed to collect the modern algae. But they don't grow in culture, so you can't just like order them up from a culture collection and work on them in the lab. You had to collect them in the field. And I called up everybody in the world who had some record of it being in the waters near them, and no one could tell me where to collect it except for this one guy who had seen it in Friday Harbor.
What?
In the 80s.
In the 80s.
I'm not kidding.
Okay.
So I told my PhD advisor I have to go to Friday Harbor, Washington, and he's like, okay.
Wow.
So long story short, I find it in Friday Harbor. And I was trying to describe the species, and I did stuff like characterize the chemistry of it and the cell wall, but I needed to just say what species I was working on, and I realized it was a different species than the ones described. So it was a new species, and I'm not like the kind of scientist that typically describes new species, so I was like, why hasn't anyone described this before? It was kind of a pain for me. I was asking around, hasn't anybody worked on this before? And people kept saying, oh, there was this guy named Maurice Doobie, who was a professor at Western in the 70s and the 80s, who worked on it.
Wow.
But he never published anything because back in those days, Western was more of a teaching college and professors weren't necessarily publishing.
Right.
And
is he still around?
He died tragically in a bicycle accident on his way home from campus one day in 1989. So there's sort of this tragic history. And all of this stuff seemed to disappear. And I was a grad student in like 2005 or 6. I called the biology department at Western. And which is funny now because these are the folks that I work with.
Right.
I probably called up Peter and I was like,
where's his stuff?
Where's his stuff? Do you have any? And everybody at Western was like, no idea, nothing. So I was really sad and I was like wondering, what would Maurice Doobie say about this algae? I need to describe the species. And I gave a talk at a conference and someone came up to me after my talk and she said, I recognize this algae is the one my friend Maury worked on. And I was like, whoa, Maurice Doobie, you knew him. It was like my first link finally to this guy.
The Doobie spheres.
And she was like, he was my friend and I have all of his stuff.
What?
And then she's like, I want to give it to you. So she mailed me these 2 big cardboard boxes that had all of his research materials that had originally come from Western. And so then I received these back in Cambridge and I took and they were like these time capsules. They were amazing. And this is where the art part comes in. Maurice Doobie was an artist and he, in order to describe these things as a new species, besides the DNA, they have these little like intricate scales that are like nanometer size. They're super small. Algae usually have different parts of their life cycle and this is like a little swimming part and it's covered with scales and all these different kinds of scales and they're really intricate. And it's really hard to do that kind of microscopy. I tried and I failed. Like you have to be an expert and he really was an expert and an artist in that and he had drawn all of these beautiful pictures. And I found out later that he taught a biological illustration course at the biology department at Western. For years.
That's what I'm trying to say.
Which is a course we do not teach anymore.
Right. I feel like it's this lost art that people don't have. We do have a science and art like minor, and I wonder if that's something that we need to talk about that afterwards. Maybe we can talk about that.
Yeah. Anyway, that's a really cool story, and that kind of connected me to Western way before I had any idea I would be a professor here.
If there was somebody out there that's really interested in what you're saying, like really concerned about what you're saying and more algae in the mountains, there is some citizen science that you do, right?
Right. So it takes a long time to climb a mountain. So we went to my lab group went to Mount Baker this weekend, but we were basically on one side of the mountain. We only had the ability to sample like one particular area. What we want to do is we want to sample as many places as possible because we want to know how many places snow algae are blooming and when they're blooming. And then once, we want to know that that's happening. And then we also want to know who's in that bloom, how diverse is it, and if it's related at all to some
Right.
of the other things that we've sampled. So to do that, we've created these citizen science collection kits, and we're kind of calling it climber science or hiker science, because we are recruiting people that are getting out into the mountains to take our little sample tubes with us and scoop pink snow into them and then mail them back to us. And that's going to expand the number of places that we can sample this summer.
Yeah,
And hopefully going into subsequent years, because in order to ask these questions about how climate change might be affecting these communities, we have to watch them change over time.
Right.
So if me and my lab group can only be in a few places, at once, or we can only sample so many places in a summer.
Right.
If we get all of these other groups to help us sample, then we're going to be able to get way more samples and get closer to accomplishing our goals.
Yeah.
So
I mean, I personally love citizen science projects. So for our listeners and viewers that maybe not know this term, this idea of citizen science of, basically anyone can go out and, take your kit and go to the mountain, collect that snow and send it to you. But there's other things, like there's I think Zooniverse, which is the astronomy citizen science website where you can go and you can help astronomers categorize galaxies and all that kind of stuff and look at images. So there's so many things that people can do. You don't necessarily have to have, PhDs or any sort of science degree or go to Harvard.
No, not at all. This is great. And I also it's appropriate for anybody. So folks that are just going on little hikes with their kids and it's early season and you see snow and you see pink snow patches and everyone's like, why is the snow pink? That's a great chance to take a sample. And we're also recruiting guide services and groups like the mountaineers that are going out and climbing big mountains to go get samples and higher up on the mountains that are hard to get to.
Okay, that's awesome.
Yeah, so it is really awesome.
Could people just go to your website, the Kodner lab, and
we have a
ask you?
citizen science tab, and it says everything about what we're doing and how to get a kit. And we've been putting calls out for citizen scientists through different social media outlets and posting on web boards for backcountry skiers. And folks have been super responsive. I'm really excited.
Awesome.
So I'm sending out kits tomorrow to the first round of people. I have about 30 kits I'm sending out, and people seem to be really excited. And I think a lot of people who are already getting out into the mountains, I've gotten a lot of responses from people who, maybe have a PhD and they're not doing science anymore, or responses from people some parents that are homeschooling kids and they're like, I'm looking for awesome things to do with my kids.
Right.
Or people who are just I really love science and I've been always curious about what the pink snow was, and so I'm really excited to be able to contribute to something while I'm also doing my climbing trip.
Is there anything I didn't ask you that you wanted to share with our listeners and our viewers about kind of your work and how they can kind of embrace science and not be afraid of it?
Oh, wow. I don't know. I guess maybe I could say science is all around us. I study things you can't see, and so when I look around at the ocean or at snow, or I see the ocean as a living thing because it's filled with cells that are doing things, and I look at snow and it's a living thing, and it's just that I would say that science just is all around us, and just being curious about the world sort of leads you into being a scientist.
Awesome. And so I just think it's amazing that you have this connection with astrobiology and you have this connection with bioinformatics, and you're kind of doing all this work that is very intersectional and interdisciplinary, and I guess intersectional maybe is not the right word. That's more of the work we do.
I think it could be considered intersectional.
Okay. But you're just doing so much, and I just want to thank you for coming here and telling us about it and teaching me all about biology, which is something I know nothing about. So now I know a little bit about it. So thank you.
Thank you. It was really fun.
All right. Thank you for tuning in to Spark Science, Ski2Sea Algae. This show was produced in collaboration with Western Washington University and KMRE. If you're interested in citizen science and collecting snow samples, go to kodnerlab.wordpress.com. Thank you for watching.